Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR NET exam life science question paper. This is this was conducted in March 2025. This is the shift 1 and I will be discussing the part B questions. As I have told earlier, I have not discussed evolution, ecology, immunology and all in part B. But the others I have discussed, like cell biology, molecular biology, cell signaling, biochemistry, then applied biology, methodology, plant physiology, all these I have done. So coming to the next question. So what does that say? The dominant and recessive alleles of a gene capital A, small a respectively. Okay. In 1000 offsprings, if 500 are small a and 500 are other genotypes. Okay, so they can be capital A, small a or capital A, capital A. That is what is meant by other genotypes. Which one of the following is most likely combination of parental genotypes? Now out of these, which is your parent? So take the first option, capital A, small a, capital A, small a, cross them. These are the gametes and these are the offsprings, and you get it in the ratio 1:2:1. So it is given 1000 offsprings, 250 will be capital A, 500 will be capital A, small a, and small a will be 250. But it is given in the question 500 are small a and we get 250 as small a, so that cannot be the parental combination. Now take the second one, again it is capital A and capital A, small a. Here there is no small a at all after crossing, so again not correct. Then third one, if you take you get capital A, small a and capital A, small a in 500 numbers. So that must be the most likely answer. Okay, so third must be the answer. You get 500 as other genotypes. So capital A, small a. Okay, other genotype. Whereas 500 as small a, again correct. Now if you take the fourth option, I have done it over here. Capital A, small a, again there is no small a at all. So that cannot be the answer. So third is the most likely answer. Then next one, live vaccines are generally used to protect against which of the following virus? It is your smallpox. Hepatitis A, rabies, it is inactivated virus, whereas hepatitis B is recombinant virus. Then which of the following molecular marker techniques uses single primer in a PCR reaction for identifying polymorphism between genotypes? So it is your RAPD. RAPD use single primer, non-random primers are used. SSR, AFLP, SCR used two primers. Then which experiment would best validate the bioremediation potential of a microbial strain degrading oil spills in seawater? That is, whether this microbial strain, whatever you have discovered, is good in degrading oil spills in seawater. That is bioremediation. So which experiment will help in validating that this particular microbe that you have isolated is good in degrading oil spills? Now the first option what they have given is using seawater without oil contamination, and you monitor microbial growth. You can see only whether the microbe grows in seawater or not, but you cannot tell that the microbe is degrading oil spill. There is no oil at all, so that is not correct. Testing microbial growth in seawater with added nutrients but no oil. So you add nutrients and give in seawater, the microbe will happily grow there, but there is no oil there. So how can you tell it is degrading oil spills? Not possible. So that is also not correct. Monitoring oil concentration in a system inoculated with microbes versus an uninoculated system. Okay, that will be good one, because You have a system where oil is added to seawater. You inoculate it with the microbes and you find out that yes, the microbe is degrading the oil. It is good. Whereas you have another system also where microbes are not added. Okay. And you monitor whether the oil concentration in the seawater is getting degraded. So you find that it is not getting degraded. Your particular microbe is the one which is involved in degrading the oil spills in seawater. So 3 will be a better choice. Comparing microbial growth in aerated versus non-aerated seawater sample is only to check the growth of the microbe, whether it requires oxygen or not for its growth. So that is what you will get there, not whether it is degrading oil or not. You will not get there. Then next one, hormone pregnancy test works by detecting the presence of the hormone human chorionic gonadotropin using immunoassay. Which type of cells will develop to produce HCG in pregnant female? That is your, so they have asked which cells are involved in the production of human chorionic gonadotropin in pregnant female. So the cells which are involved in the production of HCG is your trophoblast cells. That is option 2. Then which one of the following techniques can identify acetylation on a lysine residue of a protein? So it is your mass spectrometry, because mass spectrometry, it can identify acetylated protein as well as the acetylation site also can be identified by mass spectrometry. The next option is SDS page, that is only for separating protein based on their size and it has nothing to do with acetylation. Then light scattering, it will determine only the size of your protein, not whether it is acetylated or not, will not be detected. Then CD spectroscopy, again it analyzes only the secondary structure, not whether acetylation is done or not, they will not analyze. So it is mass spectrometry, that is the answer. Then next one, a transmembrane receptor protein X and a transmembrane protein bound to the actin cytoskeleton Y with fluorescent tags were expressed in a cell. So you have taken a transmembrane protein, that is X, it is free to run, and you have got a transmembrane protein Y which is bound to actin cytoskeleton. Both are fluorescently tagged. Okay. Now after doing this fluorescence tagging to these two proteins, then you conduct the FRAP experiment. That is a fluorescence recovery after photo bleaching experiment was performed, and you check which one will be the one that will reach the final destination upon conducting a running race. So okay, so naturally your answer will be the transmembrane protein X will reach the destination earlier than the Y, because actin is Y is bound to actin cytoskeleton. So it will have difficulty in running in the membrane. Okay, now I will come to the technique. Now what is this FRAP experiment? So here is your plasma membrane. It is labeled, and now before that I will tell you FRAP, it will measure the diffusion rate of the protein in the membrane. Okay. Now if the protein is attached to some other structure, such as here it is attached to the cytoskeleton, then this FRAP technique can find out whether the protein will diffuse faster or protein will diffuse slower because of the presence of this structure attached to the protein. So all these can be studied by using FRAP. Now what is the FRAP technique is that here you have the fluorescently labeled proteins on your plasma membrane. Then you bleach a certain region of the plasma membrane with a strong laser beam. Then you see for the recovery. Recovery is whether this fluorescent is recovered in the bleached area. That means if the fluorescence is recovered means the protein which is present over here has diffused into this bleached area. Okay. Now how fast the protein is diffusing also can be studied using the froth. If it is diffusing slowly, that means it is attached to some structure. That is why it is diffusing slowly. If it is diffusing freely means it is not attached to anything. It is free to move and it will diffuse faster. So here suppose this membrane has got X and Y proteins. You know X is free to move, Y is bound to actin cytoskeleton. It will diffuse slowly. Then you bleach with the laser beam. This is the bleached area and you find that X has appeared in this bleached area very fast. Okay, because it is not attached to any other protein. Whereas Y, it is bound to actin cytoskeleton. So it will slowly diffuse into this bleached area and it will result in the recovery of the fluorescence. So the options: fluorescence of both proteins will be recovered at the same rate. No. Fluorescence of X will be recovered later than Y. No, X will run and come fast before Y because it is not bound to actin. Then fluorescence of Y will be recovered later than X. Of course, that is correct. So the answer here is 3. Being membrane proteins, no recovery of fluorescence in experiment. That is also not correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe.